Hello everyone. I'm going to give the sort of opening talk here to try and set the scene a bit for the day. So we've got a lot of talks coming about some specific aspects of fungi. So I'm going to be giving a bit of a broad overview about fungi, structures, roles, relationships and knowledge. Okay, so the first question I want to address is where do fungi belong? And I really think to most people fungi are a real mystery. What do we do with them? Where do we put them? Where do we belong? What are they doing? So the first really important thing is that fungi belong in a separate kingdom of the natural world. And I think this is a really critical thing to understand, and it's quite a simple thing. I think most people with any kind of interaction with nature or knowledge of biology sort of understand the concept of kingdoms. We've got plants, we've got animals, we've got fungi, and they're up on that kind of level. So for a long time, fungi have been kind of shoveled away in the bottom of the plant kingdom somewhere, tucked away as lower plants or cryptogams, all these kind of hidden words and, shuffled aside. But fungi are a separate kingdom. There is a whole kingdom that's just fungi. And the reasons that they're separate, and there's a few words and things in here, but they're heterotrophic. They get their food from somewhere else, and that's a fundamental difference from plants, which are photosynthesizing, taking the carbon, using the sun, and getting energy through photosynthesis. Fungi must get their food from somewhere else. They do that by slobbering over things. So fungi produce extracellular enzymes. They don't have a stomach, they don't have a digestive system, it's all happening outside. So you can see already that there are really fundamental differences in the way they're doing things. They've got chitin in their cell wall, and some of you might know that that's a substance that's in insects. So really fundamentally differing from the cellulose that makes up plants. There's a couple of sort of technical things down at the biochemical level, the way they synthesise glycine, the little pointy bits on their mitochondria, they're really different. And in fact, mushrooms are closer to us than they are to Myrtaceae. So fungi kingdom is more closely related to the animal kingdom than it is to the plant kingdom in the evolutionary terms. So not only are they not plants, they're nowhere near plants. In terms of their construction, the two kind of key bits that make up fungi are the hyphae and the fact that they reproduce by spores, and I'll go into that in a little bit of detail. And the key thing about fungi is they're really diverse. So it's a whole kingdom that contains fungi in all sorts of different shapes, sizes and colours and different roles and so on. So there's a lot of diversity there. Now, I'll just mention briefly that there are some what we now call fungoid organisms in other kingdoms, and this kind of does confuse things a little bit, but the slime moulds, which have traditionally been thought of as fungi because they have a stage with powdery spores, also have a stage that's a bit like a big amoeba. So really they belong in another kingdom called the Protista, which has got sort of protozoan-like organisms, amoebae and so on. But they're often looked at by mycologists and field naturalists with an interest in fungi, but they don't belong in the fungi kingdom. And the other group of fungoid organisms that are not true fungi are the water moulds, and an important species in there, or important group, is Phytophthora. So in fact the water moulds are more closely related to brown algae in another kingdom called the Chromista, and they're just algal-like relatives of the brown algae that have lost the ability to photosynthesise. Most of the algae belong in the true plants, but the brown algae are in this other kingdom. So these are sort of little satellite groups that have always been dealt with by mycologists and treated as fungi, but they don't belong in the fungal kingdom. Now, hyphae. Now, I'll just put this rather complicated diagram up because I really like to see pictures of fungi like you would see in a textbook. So many of you will be familiar with a textbook picture of a cell with a nucleus in the middle, and if it's a plant cell, there'll be chloroplasts and mitochondria, but fungi are really different inside. The hypha is a terminally elongating cylindrical tube that just keeps growing, and also put out side branches. And there's all this amazing organisation going on at the ultrastructural level of the way that this wall, which is kind of quite fluid, it's almost like a plastic end that's fluid. All the bits and pieces that make up the wall are all assembled and transported in microvesicles and different ways. The wall itself is chitin fibres in a glucan matrix. So again, quite different. You might have seen cross-sections of plant walls with xylem and phloem. So the point of this is just that fungi are fundamentally different, really different to the way plants are put together. And that construction allows them to do things that plants can't do. So hyphae have apical elongation and also the ability to branch, and anastomose, which is two hyphae can come together and fuse. They're very small. They're in the range of 2 to 20 micrometers in diameter. Some hyphae are a bit bigger, but basically they're very small, and it's a microscopic exploring machine. And the key thing is getting this concept that it's really, really small. And I'll go into that in a minute to just sort of try and show you how small they are. So these tiny filaments are able to explore the substrate, whether it's a piece of rotting wood or in the soil or in a plant leaf. We can't see hyphae except under a microscope. What we can sometimes see is the mycelium, the aggregation of hyphae. So sometimes if we peel back a bit of bark off a rotting log, we might see this weft or web of threads. Now these are not the individual hyphae here, these are actually rhizomorphs, so that's many hundreds of hyphae all packed together to be able to see that. And all this sort of thin cottony layer is the mycelium, the mass of hyphae. The same when you're digging in the soil, sometimes you'll see this white kind of cobwebby-like fibres. We can grow fungi on nutrient media in a petri dish, and that's another way of visualising that mycelium. But I think a big issue with fungi is that you can't see that mycelium most of the time. So any handful of soil anywhere on the planet, there's going to be fungi in there, but we can't immediately see them in the same way that we can see plant roots easily. When you pull a plant up, the roots are sitting there, they're obvious. Spores are another of the characteristics of fungi, and again the key thing here is that they're really tiny. They're in the range of 5 to 20 micrometers in diameter. Some groups, like some of the arbuscular mycorrhizal fungi, have giant spores that are as big as, say, 1/10 of a millimetre that you can actually see just with a hand lens. But most fungi have really small spores. We can visualise that through the spore print in a mushroom. It's a great way for everyone of all ages, but particularly kids, of actually showing these millions and millions and millions of spores that drop down from the cap of the mushroom. That's one way of visualising spores. Puffballs are another really good way of visualising spores. You kind of knock on the puffball and a puff of powdery spores come up. And there are millions and millions of spores just in a single puffball because they're so tiny. Now this looks like a bit like a mathematics class, but I have to put this up to remember all these figures. So I want to sort of give you an idea about how small fungi are. So the first comparison I want to make is between fungal hyphae and a plant root. So if we imagine this is a plant root, so an average plant root might be 0.5 millimetre in diameter or so, and I've blown this up by 200 times. So now my plant root is 10 centimetres across. Now a fungal hypha is 5 micrometres in diameter. Now the key thing is that there are 1 million micrometres in a metre. So a micrometre is 10 to the minus 6 of a metre. So my fungal hypha that's 5 micrometres in diameter, when I blow it up 200 times, it's 1 millimetre in diameter. So this is a plant root, just an ordinary, not particularly big root of a plant, like the fine roots of a plant. I'm not talking about a big taproot or anything. So there is the plant root, and there is the fungal hypha. And this is the comparison when you're thinking about the soil. This plant root is big, clunky thing that's trying to push its way through soil particles and so on, and it's a very inefficient way of getting into the nutrients and exploring the soil. This fungal hypha has the ability to grow in between the grains of sand and the particles of soil and really explore for nutrients. And because of the ability of fungi to grow, to branch, to ramify, there are very effective means of exploring substrates. So this size comparison, I think, is a really critical thing to understand with fungi, because we see the fruiting bodies of fungi in the forest, and they're the visible bits, and they're made up of millions and millions of very fine hyphae as well. But once we pull that up, it's not like a plant. When you pull up a plant, you can the roots come up with it. the fungus fruiting body just snaps off, and we don't see that ramification of the hyphae and mycelium in the soil. So I think fungi are really small. So the way we interact with fungi is mostly through fruiting bodies. So a fruiting body is an aggregation of the hyphae growing up from a mycelium, which is in the substrate, such as a rotting log or some soil. And this is just lots and lots of hyphae packed together very tightly in a sort of defined form. And the function of the fruiting body is to produce the spores and reproduce. So they're kind of analogous to the flower of a plant. And the important thing to remember is that the vegetative part of this fungus is in the soil or in the rotting wood, and it's often persistent, and it's just sitting there. And we can't see that in between. We only kind of know the fungus is there when the fruiting bodies come up. Fruiting bodies can be quite small, such as in this rust fungus, and at a much smaller level there are lots of moulds and microfungi that don't really produce large fruiting bodies. The spores are just produced directly on a mycelium. So this is a life cycle, and even with my students at university, I say, don't worry too much about the details, but what I want you to take away from this again is that fungi are different. So the way that the spores germinate into a haploid mycelium, the way that the different parts of the reproductive cycle are separated, there's a very extended haploid phase. The true diploid phase is very, very short, and there's a special thing in a lot of fungi called a dikaryon, where the two nuclei sit together separately. These are all fundamentally different ways of reproducing than in animals or in plants or in protozoa or in other kingdoms of life. It is a marvellous life cycle if you're interested in getting to know about fungi, but it's really different. Now, sex in fungi is really different as well. So sexual differentiation is rare. So unlike in animals and in plants, there's no male bits or female bits. And sex in fungi is through mating types, and there's no differentiation there. So basically in the fungal disco here, all different fungal individuals wandering around, and what determines sexual compatibility there is not there's no secondary differentiation. I can't tell male and female. It's basically like having numbers on your back. And some fungi have just two numbers, mating types. Some fungi have hundreds of mating types. And successful reproduction is through just difference. So a 1 and a 2 are compatible, but 2 1s aren't. So in many fungi, there are lots and lots of different mating types. And this promotes outbreeding and increases the probability of finding a successful mate. Fungi also have the ability in some life cycles for siblings to interbreed. So again, there's a really different kind of a life cycle here. Asexual reproduction is extremely common in fungi, and a lot of fungi have a life cycle where there's a sexual part of it and an asexual part. This has big implications for things like plant pathogens, where there may be a stage that rotates on a crop while the crop's growing. There's lots of spores produced that enable reinfection. Then there might be a resting stage that can sit around for a long time to reinfect the crop in the next season. Or maybe it grows on an alternate host. So some fungi have a life cycle where one part of the life cycle, particularly rust fungi, is on one host, it may be a grass, and another part of the life cycle might be on a shrub. So there's some quite different things going on there, again, with fungi. Now, food. I think food's really important with fungi because it gets to the nature of what are they actually doing in ecosystems. So I use this analogy of they're just slobbering on things and then absorbing their food, and that's because they have extracellular enzymes. So fungi have a whole battery of different enzymes that they have to excrete onto the substrate to break it down. And they have a battery of enzymes for things like not only carbon, but also nitrogen and phosphorus. So wood is just something that doesn't decay of its own sort. Wood just sitting here, it will sit here for centuries. It doesn't decay on its own, it doesn't oxidise or anything, it just sits there. What's in wood is very, very tough, complex compounds like lignin and cellulose, and they're very resistant to attack by other organisms, and it's really only fungi that have the battery of enzymes that can break down the lignins and the cellulose release the carbon, get the nutrients cycling through an ecosystem. So organic compounds of all different sorts are really yummy for fungi, and fungi decompose not only wood but a whole range of other organic compounds, including compounds in fingernails, feathers, stuff like polyurethane, kerosene, any kind of organic compound. There'll be a fungus out there that's got an enzyme that can bust it apart and use that for food. Fungi are also really important for nitrogen and phosphorus, particularly in Australia's really impoverished soils, very low nutrient, highly weathered and leached soils. So nitrogen can be present in the soil, but in a form that a plant can't take up, and the fungi are able to get in there and change that nitrogen around, either absorb it themselves and pass it on to the plant, or solubilise it, or make it more available for a plant. So these enzymes are important for the fungal nutrition, but they're also getting substrates that are not available to other organisms into food webs. So decomposition is really important, and fungi are these primary decomposers of dead wood, and then the fungi themselves become food for invertebrates and so on in the food webs. So I think it's always great to find a nice bit of rotten wood, particularly in a wet forest, and the amazing thing is just to be able to put your finger into that wood, straight through the wood, where there's fungal activity. You can see here the fruiting bodies of this psilocybe, and here are the rhizomorphs and the mycelia ramifying through the wood. And some huge fallen trees in a rainforest, you can just punch your finger straight into that because of the fungal rot. The intact wood will be as hard as this, but the fungi's got the ability to get in there, chomp up all that lignin and break it down. So there's a number of different trophic strategies. Trophic just means food strategies that fungi have. So they have to get their food from somewhere else, so they're heterotrophic, but they can do that in a number of ways. So different groups of fungi do this in different ways. Many fungi are decomposers or saprotrophs. They're just taking already dead stuff that's sitting around and breaking it down. And if those fungi weren't there in a forest, the litter and the wood would just build up and up and up. So very important role in taking that dead organic matter, utilising it for food and recycling it. Now another way that fungi get their food is to parasitise other living organisms. So there are many fungal pathogens of plants, of animals, and also of other fungi. So that's just saying, I can't be bothered to making a big effort, I'm just going to parasitise on something else. And all life on the planet, virtually there will be some fungus somewhere that's making use of somebody else's work in getting food. But there's also a third really important way that fungi get food, and generally the mode of nutrition tends to be sort of different for particular genera or species. Some things can switch from one to another, but in general this sort of way of getting food is different for each different fungus. And mutualism is another really important way. So this is a partnership between two organisms where they're swapping nutrients or something else to their mutual benefit. So it's a kind of symbiosis, but it's a particular kind of symbiosis where there's a mutual benefit. A lichen is an example of a mutualism. That's a mutualism between a fungus and an alga or a cyanobacterium. And mycorrhiza is another example of this kind of mutualism. And these interactions and networks, fungi are very much involved in interacting with other organisms. So in a mycorrhiza, which is really just like a fungus stock, this is a cross-section through a root of a plant, and here's the fine hyphae around the outside of the plant root that form a kind of definite structure around the outside of the plant root, and there's an exchange of nutrients. The plant is providing sugars to the fungus, and the fungus is providing different forms of phosphorus and nitrogen to the plant. There's a lot of benefits here in both in the exchange of nutrients, but also things like disease protection just because of the physical barrier, and even the ability to confer heat resistance, drought resistance and so on to the plant. So these are very important interactions. And just to sort of show you what's going on there, again, it's like here's the plant root and here's the fungal stock on the plant root there. So most plants actually have this thin layer of fungi growing on the outside, or a lot of forest trees. Another way that fungi do it is to grow, penetrate in between the cells, and there are different kinds of mycorrhizas, but most plants have got some sort of fungal growth going on there. And just to finish off, I'm just going to flip through and just talk briefly about the molecular revolution and fungi. So what's really hampered us until now is being able to get at fungi. You've got all these fungi in the soil. How do we know what's there? How do we tell them apart? We've got all these different species. How do we know what they are? How do we tell them apart? And DNA sequencing, it's the kind of technology that's used in forensics, is really revolutionising our ability to get fungi out of substrates, to identify them, to tell them apart. And if we were to kind of take some soil, kind of wash away all the soil particles, that's kind of what's going on in the soil. All different fungal hyphae, all tangled up together, lots of different species, all doing different things. And we now have the technology to grab that handful of soil, stick it in a test tube and pull all the fungi out of it. And the study I'm involved in at the moment with a guy called Laiho Tedursu in Estonia, just taking handfuls of soil from all around the world. In the 50 sites in Australia that have been sampled, each site it would end up just being like a teaspoonful of soil. Just in those 50 sites we have found 3,000 different species of fungi. So that's half the fungi that are already known from Australia. Now many of those we can't identify as species, but we know that they're all different. So this molecular revolution is allowing us to get at all this fungi and start to really answer some really interesting questions about what are they doing and how do they differ from one site to another. So I'll just finish off, that's just some lovely DNA there, so DNA can be beautiful as well as fruiting bodies, the patterns in it, the way that they differ, and in essence it comes down to this difference in particular bases at particular positions in the DNA, and it's fantastic information. And so just to finish off, fungi really are networkers, they interact with other organisms, they're out there doing lots of important things, and in this Fungi Festival we're aiming for a multi-way transfer of knowledge of your interests, your knowledge, your enthusiasm, and talking about challenges and solutions. So hopefully you enjoy the rest of the day.